Hello, good morning to all of you children. Myself Dr. Devbrata Mukherjee, Kendriya Vidyalaya number 1, Kachrapada, Kolkata region. Today I would like to explain, to interact with you rather to say about the inheritance of two genes under the chapter 5, Principles of Inheritance and Variation. So friends, first of all we must know about the gist of the lesson, what we will study in the part. Exactly, we will discuss about the Mendel's law of inheritance, inheritance of two genes, and output law of independent assortment. So we must know the who is Gregor Mendel. He is said to be as a father of genetics and principles of independent assortment he discovered, and it was during the mid 19th century that headway was made in the understanding of the inheritance, and in that perspectives Mendel discovered law of the principles of the Mendelian inheritance. It is rather said here the law of segregation, law of the independent assortment, and the law of the dominance. We have already discussed these points in my previous video lecture. Let us recapitulate at the onset. So the Mendelian principles, if you would like to talk about, the Gregor Mendel, the conducted hybridization experiment on garden pea for several years, 1856 to 1863, and he proposed the law of the inheritance in living organisms. Though the target plant, his basic plant on which he did the research, that was the garden pea, Pisum sativum, and two general rules of the inheritance from their output was there. He proposed the principles of laws of the inheritance. First law, or the law of dominance. Second law, law of the segregation. So what about the law of the dominance? That is, the characters are controlled by discrete unit, that is called factors. Factors occur in pairs. In a dissimilar pair of factors, the one number or the one member of the pair dominates, that is dominant, and other is to be the recessive. These two terms will be used subsequently throughout the lecture. So the now today's topic is inheritance of two genes. How did it been proved? Mendel also worked with the crossing pea plant that differed in two characters. Number one, the cross between a pea plant that has seeds with the yellow color and round shape, and one that has seeds of the green color as well as this been wrinkle shape. What was his observation? What did he observe? The seeds been resulting from the crossing of the parents had the yellow colored and round shaped seed. Here, can you tell students which of the characters in the pairs, yellow or the green color and round or the wrinkle shape, was dominant? Let us discuss how is it possible in the explanation portion. Thus the yellow color was dominant over the green and round shaped dominant over the wrinkled. These mean results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and the wrinkled seeded plants. Capital Y is the symbol of dominant yellow seed color. Small y means recessive green seed color. Capital R for the round shaped seed. Small r is the wrinkle shaped seed. So the what was the genotype of a parents? Here the genotype is the capital R, capital R, capital Y, that is called round yellow seed. Small r, small y means the wrinkled green seed. The gametes, capital R, capital Y, small y, unite on fertilization, and that produces the F1 hybrid, round and yellow. It is the heterogeneous, that is capital R, capital Y, small y. But when Mendel self-hybridized the F1 plants, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had the green seeds. So how the segregation is possible? The yellow and the green color segregated in 3:1 ratio, and round and the wrinkled seed shape also segregated in 3:1 ratio, just like a monohybrid cross. It is partly. So two monohybrids combine one dihybrid cross. So the phenotypic ratio is there round yellow is to round green, wrinkled yellow is to wrinkled green, and the simultaneously here we could say that the 9 is to 3 is to 3 is to 1 obviously we are taking. From there the derivation was there, the law of the segregation. So how does this law was, so how does this segregation law has been developed, so the coming to the law of the independent assortment. In the dihybrid cross, the phenotype round yellow, wrinkled yellow, round green and wrinkled green appears in the ratio 9 is to 3 is to 3 is to 1. Such a ratio was observed for several pairs of the characters that Mendel studied. So the ratio of the 9 is to 3 is to 3 is to 1 can be derived as a combination series of 3 yellow is to 1 green, 3 round is to 1 wrinkle. So just like this derivation can be written as follows: 3 round is to 1 wrinkle into 3 yellow is to 1 green is equal to 9. Round yellow 3 is to wrinkled yellow is to 3 round green is to 1 wrinkled green. So the derivation has been clearly depicted here. So based upon such observation, what is the output? Dihybrid crosses concept came into there. The crosses between plants differing in the two traits. This is the definition. And Mendel proposed second set of generalization that he called Mendel's law of independent assortment. What does the law state? The law states that when two pairs of traits are combined in a hybrid, segregation of one pair character independent of the other pair of the characters. So let us explain in the light of Punnett square. The Punnett square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of eggs and pollens in the F1 capital R small r capital Y small y plants. And we consider the segregation of one pair of gene capital R and small r, 50% of a gametes of a gene capital R, other 50% of small r. Now, besides each gamete having either the R, capital R or small r, it should also be allele capital Y or small y. So the important thing to the segregation of the 50% capital R gene, 50% small r gene, and the is independent from the segregation of 50% capital Y, 50% small y. Similarly, 50% of capital R bearing gamete has the capital Y, other 50% has the small y. 4 genotypes of a gametes, 4 types of pollens, 4 types of eggs are formed. The 4 types are capital R, capital Y, capital R, small y, small r, small y, small r, the small y, each of which frequency of 25% of the 1/4 of a total gamete produced. So what will write down the first step of the eggs and pollens on the two sides of a Punnett square, which is very easy to derive the composition of the zygote that give rise to F2 plants. In the table it is depicted. So let us discuss a common answer of a common question. From there to check our understanding. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. You find out. And the next one, can you using the Punnett square data work out of a genotypic trait, at least the genotypic trait at a F2 stage, fill in the format given. Is the genotypic ratio is 9 is to 3 is to 3 is to 1. So at last, I have been placed some assignments. You just do and find out the answers, and now the references you follow, and some YouTube links I have placed here, and some YouTube links, videos made by me and from other sources, inter-syllabus YouTube links, and different chapter wise the website links are there. That's all. Thank you very much.